Good afternoon, everyone. Welcome to another video lecture from us on DNA's Learning Center. Today, I'll be teaching you a lesson on chitin, its structure, specifically N-acetylglucosamine units, and its crucial functions in exoskeletons. We are Group 1, and our members are Koreen, ACOSTA, Roxelle, ANCAJAS, Joshua AUGUIS, and me, your lecturer, Clint AMODIA, all coming from BSMT 1D. To begin, let me ask you, have you ever wondered what makes an insect's shell so strong yet incredibly light? Today, we'll delve into the fascinating world of polysaccharides found in nature and introduce you to chitin, a remarkable structural carbohydrate. We'll also go over our learning outcomes and provide a brief overview of what we'll discuss. By the end of this short lesson, which should take about 9 to 15 minutes, you should be able to define chitin and identify its monomeric unit, describe the chemical structure of N-acetylglucosamine, and explain how it forms chitin. Explain the biological function of chitin in exoskeletons and other organisms, and finally, appreciate the structural importance of chitin in nature and its potential applications. Let's start with the definition and occurrence of chitin. Chitin is a structural homopolysaccharide. This means it's a large molecule made of repeating identical sugar units. Specifically, it's composed on N-acetylglucosamine or GLC-NAC units linked together by what are called beta-1 for glycosidic bonds. So, where do we find chitin? It's widely found in nature, most notably in the exoskeletons of arthropods like insects and crustaceans, the cell walls of fungi, and even in the beaks of cephalopods such as squids. Now, let's zoom in on the building block of chitin, which is N-acetylglucosamine, or known as GLC-NAC. It is the monomeric unit of chitin, and it is essentially just a modified sugar, structurally quite similar to glucose. What's unique about it is a chemical modification, which is the hydroxyl group on the second carbon of glucose is replaced by an acetylated amino group, which is NHC, OCH3, and this subtle change is crucial for chitin's properties. Its molecular formula is C8H15NO6 in terms of functional groups. It has hydroxyl groups on the most carbon atoms, just like glucose. But the key is that acetamido group on C2. This group provides both polarity and the capacity for hydrogen bonding. The ring form is a 6-membered pyranose ring, similar to glucose. The importance of acetamido group cannot be overstated. It enables extensive hydrogen bonding between adjacent GLC-NAC units, which is key to chitin's strength. It also contributes to the rigid crystalline structure of chitin fibers and makes chitin more resistant to enzymatic degradation compared to cellulose. You can think of GLC-NAC as a glucose molecule wearing armor. These acetyl and amino additions make it tougher and more specialized, perfect for building protective structures like insects, shells, and fungal walls. Moving on to these monomers, which come together to form chitin. Chitin is a linear polymer. This means the GLC-NAC units are arranged in a straight chain, connected by those beta-1 for glycosidic bonds we mentioned, much like cellulose. The polymerization happens through a dehydration synthesis reaction, where a water molecule is removed as each pair of sugar units joins. Crucially, the acetylated amino group on the second carbon of each GLC-NAC monomer, instead of hydroxyl group, significantly increases its hydrogen bonding capacity. This allows adjacent chains to align very closely, forming tight bundles called microfibrils. These microfibrils are incredibly strong and insoluble in water. They can also be further cross-linked with proteins, for instance, in insect cuticles, which can enhance either flexibility or rigidity depending on what the organism needs. Ultimately, these microfibrils provide strong resistance to mechanical stress and offer protection against environmental hazards. Now, let's explore the diverse biological functions of chitin due to its rigidity, flexibility, and biodegradability. Chitin plays vital structural and protective roles across various organisms. In arthropods, such as insects and crustaceans, chitin is the main component of their exoskeleton. This exoskeleton provides essential mechanical strength and serves as an attachment site for muscles, enabling its movement. It's remarkable how this exoskeleton is lightweight yet very durable, aiding in both protection and also in mobility. In fungi, including yeast, molds, and mushrooms, chitin is a major constituent of their cell walls. It's crucial for giving the fungal cell its shape and helping it resist osmotic pressure. It essentially acts as a support scaffold, sometimes embedded with other polysaccharides like glucans. Chitin is also found in mollusks and cephalopods such as in squid beaks and radulae, which are rasping tongues, providing them with stiffness and cutting ability. In nematodes and parasitic worms, chitin lines parts of their egg shells or cuticle, playing a role in their reproduction and environmental survival. Even some bacteria and algae incorporate chitin-like polymers in their protective matrices, forming biofilms and microbial mats. Beyond these structural roles, chitin and its derivatives also have other functional highlights. For example, chitin derivatives, especially chitosan or chitosan, have shown inhibitory effects on bacterial growth, meaning they have antibacterial properties. And unlike plastics, chitin is biodegradable, meaning it can be naturally broken down, making it useful in sustainable material development. Now, we are on the last, summarization and generalization. To summarize, we've discussed the definition, structure, and functions of chitin. We learned that chitin is a crucial structural polysaccharide, providing strength, protection, in diverse organisms. Its unique chemical structure, particularly the acetamido group on N-acetylglucosamine, allows for extensive hydrogen bonding, leading to its remarkable properties of rigidity and insolubility. We also touched upon its biodegradability and other properties that make it relevant for various applications beyond just biological structures. We hope you now have a clear understanding and appreciation for this important biomolecule. If you have any questions for us, we'd be happy to answer them down on the comment section below. This is it for Chitin, this is it for me, and I hope that we can still see you on the next lesson. Bye bye, peace out.